Hi! In this video, we present a full single quantum dot tracking experiment.
A dedicated algorithm has been entirely conceived in collaboration with image analysis experts.
During this video, we will use the epidermal growth factor receptor as a model to describe the method of this technique. The epidermal growth factor receptor is a transmembrana protein. This receptor will be tagged using a monoclonal antibody which is reduced to keep only the FAB fragment. The FAB fragment is biotinylated and after that linked to the streptavidin of the quantum dot. The whole system will precisely recognize the EGF receptor.
Our goal is to provide a comprehensive view of cell surface receptors dynamics. Indeed, molecular trajectories can deviate from Brownian diffusion by being confined within nanodomains, for instance, and this may be the signature of an underlying interaction with, for example, signaling partners or molecular scaffolds. We aimed at describing such events exhaustively by mapping them over the cell, which requires working at high spatiotemporal resolution together with high labeling density. We hence named this approach multi-target tracing.
The first step of the experiment is the preparation of the sample. We work on live cells without antibiotics. Here we use COS7 cells, which endogenously express EGF receptors. After detaching the cell, we need to count them precisely in order to have the same number of cells in each well of the lab tank. A precise number of cells is very important to increase the reproducibility.